Welcome to our final subtopic for all of senior biology. I can't believe we're already at this point. All we have to go through now to reach the end is looking at pushing that idea of evolution a little further along. We looked at, well, what is evolution? We looked at microevolutionary changes through things like allele frequency. In this video, what we're going to go through is speciation and macroevolution. So what happens when you have an accumulation of these microevolutionary changes? We have enough changes in the gene pool that we end up with brand new species. So these are all the objectives we have to go through in this subtopic. It's going to be split up into two, maybe three videos. And in this video, we're just going to be focusing on those first two there, looking at speciation and how that results in change over time, and also looking at mechanisms of isolation, which will become more important as we go through. So everybody remembers the famous example of the Galapagos Islands and Darwin on the Galapagos Islands. And it wasn't just about finches. There were also the giant tortoises there, which all evolved from a single common ancestor, much like the finches, but they diverged to produce multiple different species. So seen here, we've got three different species of Galapagos tortoise. You've got the Pinta Island species, which has, as it says there, an intermediate shell. You've got the Isabella Island species, which had a dome-shaped shell. It's hard to say. And then the Hood Island species, which has a saddle-backed shell. Now, each of these shell types are going to be as a result of selective pressures on these respective islands. For example, you'll see that the Hood Island species has quite a long neck, and that saddleback shell allows it to extend its neck up and reach food that is higher up. So that's an adaptation there, thanks to natural selection. So when we have a single species like this evolving into multiple species, that's a process known as speciation, and that is our macroevolution. So speciation is the evolutionary process by which populations evolve to become distinct species. Okay, so we should remember from early in Unit 3 when we defined what a species was, and there are multiple definitions of it, but the biological species concept says that two organisms are of a different species if they are incapable of reproducing to produce fertile offspring, or conversely, two organisms of the same species must be able to successfully interbreed. Okay, so that's the definition of a species. And if we undergo speciation and macroevolution, then what we're going to have is organisms that can no longer reproduce. Going back to our giant tortoises before, those three species of tortoise can no longer reproduce because they are completely different species. They are what we call reproductively isolated from each other, meaning that they are unable to reproduce. We're not necessarily talking about physical isolation. So that this biological species concept, that two different species can't reproduce, is the one that is used widely in evolutionary biology. It talks about species as being genetically or reproductively isolated groups, and that allows us to contextualize and understand speciation, etc. There are multiple different species concepts, and there are exceptions to it, which you should revise as part of your Unit 3 study. So if we have a look here, how does microevolution lead to speciation, etc.? Well, this is all happening over time, and it's all to do with an accumulation of changes. So we know that microevolution is when natural selection favors certain phenotypes that are better adapted, natural selection. Okay, we remember that. And that leads to changes in the allele frequencies of the gene pool. Now, if that builds up over time and we end up with populations that become increasingly different in terms of their gene pool, then we end up with speciation. So there's enough microevolutionary changes building up that it results in a new species. They've now changed so much, they're reproductively isolated. The end result of all of this is macroevolution. which is when speciation events lead to the development of a collection of new species, genera, families, higher taxonomic orders as well. So speciation is the result of lots of microevolution, and macroevolution is when we're looking at how a group, a taxonomic group, has evolved through speciation events. So reproductive isolation is the key here. How do we get reproductive isolation? That's what we want to know. How do we get to that point? So speciation, as we know, is that reproductive isolation, and it's when a single population over time becomes two separate populations that can no longer breed. And that's our reproductive isolation. So the original gene pool that we had is divided, and due to different environmental conditions, say, the selective pressures through microevolution will act differently on those populations, and will continue to act differently on those populations until the allele frequencies are so different that they become completely new species. So we have an accumulation of those microevolutionary changes. So think about, a population of frogs, and all of a sudden there's some sort of isolating event. Maybe, let's say, a flood comes along and washes away a heap of the frogs into another area. We've now got the population of frogs that didn't get washed away, and we've got the population that did get washed away. Now, the population that got washed away got washed downstream, and they're in a completely new environment now. So as a result of that, they're going to have different selective pressures acting upon them, and they will change differently. Over time, that isolating mechanism here, which is physically separating them from each other, is going to result in different selective pressures acting on those organisms, resulting in new species entirely. So two different species from that original interbreeding one population of frogs. And they have an increased genetic distance in them, which we can track with phylogenetic trees. Our mechanisms of isolation come in one of two overarching varieties, and that's pre-reproductive isolating mechanisms, effectively stopping them from being able to mate. Okay, so pre-reproductive isolating mechanisms stop mating or sex from happening. Post-reproductive isolation mechanisms prevent fertilization. So they're not preventing mating, but they're preventing fertilization or successful mating. They could prevent the embryo from developing, or they could produce offspring that are inviable. So they can't reproduce themselves, they're infertile. Our pre-reproductive isolating mechanisms, you've got four main types here. First one is a pretty big, major, obvious one, geographic isolation. So if you've got individuals or subpopulations that are separated by mountains or seas or distance or habitat in some way, some sort of actual physical separation, then that can stop them from isolating, obviously. There are some frogs in Victoria and Tasmania, certain species of frogs that are incredibly closely related, genetic testing has shown, but they're separated by the Bass Strait when sea level rose after the last ice age. And what that meant is, because of that intense separation, they underwent different selective pressures, and as a result, they're now different species. Another example of this as well, and sort of artificial example, I suppose, is the Great Wall of China. There are certain organisms, beetles, etc., that aren't able to fly up and over the Great Wall of China. So where once they lived in that area and they were one big population, they were separated there. Temporal isolation is temporal meaning time. So they breed at different seasons, different times of years, or different time of day. And then behavioral isolation is different courtship patterns. Okay, so there's nothing physical preventing them from mating in temporal or behavioral, but it comes down to, well, when do they breed? What time, or how do they attract a mate? If you've got a bowerbird, I think it's a satin bowerbird, which is really attracted to things that are blue, and then you've got one weirdo bowerbird that's really into red, and so he builds up his bower, his nest of all things red. It's not going to bring in any of the ladies because he has different courtship patterns. And then lastly, morphological isolation. So they want to get it on, but their bits just don't match together. It's like trying to charge your iPhone with your friend's Galaxy charger. It's just not going to happen. Thanks to the Amoeba Sisters for these great images here, and you should check out their video on reproductive isolation because it's got some great examples, but these pictures are fantastic. So geographic isolation, really straightforward. An actual geographic physical separation that stops them from getting together to mate. So it's not necessarily that they don't want to or that they couldn't if they were next to each other, but they just can't get to each other. Temporal isolation, reproducing at different times, and we do actually see this with plants quite often, okay? So reproducing at different times of season, different times of day, etc. And the behavioral isolation comes down to the courtship pattern. So whether that be through singing of bird song, or whether it be through displays of dances or things like that. And courtship patterns, obviously, fantastically shown by birds. So those are all our pre-reproductive isolating mechanisms, okay? As well as that, we've got the morphological one. I'm not going to show you different genitals. I'm sure you can figure out, the differences there. So post-reproductive isolating mechanisms, as we said, they're not stopping the reproduction from happening, but they're stopping it from being successful. So it prevents viable fertile offspring resulting from that mating. We've got gamete mortality, which means that the gametes do not survive. So they can have intercourse, they can have sex, they can mate, but for some reason the sperm won't survive in the reproductive tract, or the egg won't match up with the sperm or something like that. So that's the first sort of stage of post-reproductive isolating mechanisms. If we look at the next one, okay, so fertilization has occurred. The sperm has met the egg, successfully fertilized it, but then the zygote doesn't survive from there. So something happens, it just doesn't develop any further from there. And that could be through any number of different biochemical processes. And then lastly, we've got hybrid sterility. So the offspring develop and grow, so we can actually have offspring result, but they're infertile and they won't be able to breed and pass on their own genes. And this is usually the result of aneuploidy, the wrong number of chromosomes. Let's say in the zonkey example, which absolutely adorable, a zebra-donkey hybrid. Let's say the zebra has an N of 42, and then the zebra has an N of 31. Then when they get together to produce a diploid, then they're going to have 73 chromosomes, as opposed to the zebra, which should have 84, or the donkey, which should have 62. So completely wrong number of chromosomes. They can survive, but they're not necessarily going to be successful. Okay? So that's our isolating mechanisms. That's where we're going to end it for today. In the next video, we're going to look at modes and patterns of speciation.